Okay. First of all, I would like to thank the organizers of this meeting for inviting me to present some of our data on cell therapy for epilepsy. Particularly, I want to thank Norberto and his team for doing an excellent job of managing this conference. So the topic of my presentation today is on the promise of neural stem cell therapy for counteracting hippocampal injury or status epilepticus-induced impairments in memory, mood, and neurogenesis. Before going into stem cells, I'll give a brief introduction on hippocampal injury. So as we all know, hippocampus is very important for cognitive and mood function. And hippocampal injury can occur through a number of different causes. This includes head injury, ischemia, stroke, acute seizures or status epilepticus, severe chronic stress, and early stages of Alzheimer's disease. Now, consequences of hippocampal injury. There are two phases, acute phase, early phase after injury, and then you see some changes in the chronic phase. And acute phase changes, Steve Danzer has already showed you, there's increased neurogenesis, and part of that neurogenesis is abnormal. So there's displacement of granule cells into the dentate hilus. Then you also see increased concentration of neurotropic factors. Both increased neurogenesis and elevated concentration of neurotropic factors, they're likely, the compensatory mechanisms due to injury. And then because of neurodegeneration, you also see reactive astrocytes and activated microglia and contributing to neuroinflammation in the hippocampus. Then in the chronic phase, the situation is very different regarding neurogenesis. So even though it was increased in the early phase, in the chronic phase, there's dramatic decline in neurogenesis. Even the residual neurogenesis is abnormal. And then over a period of time after injury, you do see loss of interneurons. I think Michele demonstrated that in his models. So different subclasses of interneurons, they kind of disappear. Some of that is due to actual cell death, and some of that is due to the loss of protein. And that includes reelin-positive interneurons. And then you see decline in neurotropic factors. We have actually measured the concentration of BDNF, FGF2, and IGF-1. All three neurotropic factors decline in the chronically epileptic hippocampus. And it eventually leads to memory and mood dysfunction. And depending on the type of model, you can also see chronic epilepsy typified by spontaneous recurrent seizures. Now, we asked the question, can intrahippocampal neural stem cell grafting early after focal injury avert hippocampal dysfunction? So initially, we started with the focal injury model. In this model, what we do is we inject a very small dose of canic acid into the right lateral ventricle of the brain, and that causes very specific lesion in the hippocampus ipsilateral to injection. You lose some hilar neurons and a significant number of CA3 pyramidal neurons. There's also some partial damage in CA1. And then over a period of time, different fractions of GABAergic neurons, they also decline in number. And then you see neuroinflammation, and you see increased neurogenesis in the early phase, and then greatly declined neurogenesis in the chronic phase. And part of that neurogenesis in the early phase is also abnormal. And in contralateral to canic acid administration, you don't see much changes. There is some increase in neurogenesis. Other than that, you don't see any neurodegeneration. And the overall outcome in this model is cognitive and mood dysfunction. And physiologically, if you test different areas of the hippocampus, you see hyperexcitability. But spontaneous seizures take a long period of time. You have to wait at least 4 months before you see behavioral spontaneous seizures in this model. So now I'll show you some of the changes which occur after intracerebroventricular canic acid administration. This slide, you see nuance-stained pictures here. This is control hippocampus, and this is the hippocampus ipsilateral to kinate. And if you quantify the neurons, you see loss of neurons in the dentate hilus, and also in the CA3 area here, you can see here ipsilateral to kinate, significant reduction in CA3 pyramidal neurons, and some loss in CA1 too. We also quantified interneurons using GAD67 immunocytochemistry, and we quantified in different cell layers of dentate gyrus and CA1 and CA3 subfields. In all of these subfields, you see a decrease in the number of GABAergic interneurons. It shows activity of astrocytes after injury. This is control hippocampus. This is after injury in the dentate hilus. You can see hypertrophy of astrocytes. They are reactive astrocytes. Then you also see here activation of microglia. So this is done with ED1 staining. This is early after injury. And this is in the chronic phase. You can see that inflammation decreases, but still you see some activated microglia in the degenerated CA3 cell layer. And then neurogenesis in the early phase, it increases in the entire hippocampus. We did stereology counts of double cotton positive neurons. Then we also quantified separately for the anterior half and posterior half of the hippocampus. And we see decreased neurogenesis at both, along the anterior posterior levels. And then part of this neurogenesis is abnormal. You can see migration of newly born neurons into the dentate hilus in this picture here. But in the chronic phase, neurogenesis dramatically declines. And this is for the whole hippocampus. In comparison to control, there is significant decrease. And again, this occurs along the septotemporal axis of the hippocampus. So now coming back to the question, can intrahippocampal NSC grafting early after focal injury avert hippocampal dysfunction? So we chose subventricular zone stem cells for this study. One can use neural stem cells from any region of the brain, particularly from the embryonic brain. Any region you collect neural stem cells and expand in culture, you get a large number of neural stem cells. But in the adult brain, only two regions are considered neurogenic. One is hippocampus, and the other one is subventricular zone lining the ventricles. The hippocampal stem cells, they produce neuroblasts, and they eventually become granule cells in the dentate gyrus, whereas in the subventricular zone, the stem cells produce a large number of neuroblasts, which migrate along a pathway called rostral migratory stream. And they get into the olfactory bulb, where they differentiate into olfactory bulb interneurons and granule cells. That is in normal conditions. But in conditions such as a stroke or ischemic damage, these neuroblasts take a detour. Instead of going into olfactory bulb, they get into injured regions, and they exhibit site-specific neuronal differentiation. For example, some of these neurons can go into the injured striatum, where they differentiate into medium-sized spiny neurons expressing DARP32. So they have some ability for repairing the damaged brain. But when you culture these cells, very easy to culture. So you just scrape off the ventricle wall and then use regular neural stem cell proliferation medium. They expand very nicely. You get neurospheres, and they maintain their multipotency and also self-renewal ability over extended periods of time. So that's why in this study we chose subventricular zone neural stem cells. And so it's a rich source of neural stem cells, subventricular zone, easy to grow, they maintain multipotency, and then they express a variety of neurotropic factors like GDNF, BDNF, VEGF, and so many other. They also generate significant numbers of GABAergic interneurons. So about 25% of cells derived from these stem cells, they differentiate into neurons, and most of these neurons turn GABAergic. That seems like a default differentiation of these cells in culture conditions we use. This shows the research design we employed for this study. And on this side, you see the preparation of host rats for transplantation. So we started with young adult male rats, and we injected canic acid to the right lateral ventricle to induce hippocampal lesion. It's a unilateral injury. And then 5 days after injury, we transplanted neural stem cells into the hippocampus, only to the injured hippocampus. We placed 3 transplants, each containing about 100,000 live cells. And on this side, you see the preparation of neural stem cells. We collect subventricular zone tissues from postnatal day 2, transgenic rat bulbs expressing alkaline phosphatase. So we use these transgenic rats because every cell is positive for alkaline phosphatase. That makes it easy to track these cells after transplantation in the host brain. So we expanded these stem cells in stem cell proliferation medium, got neurospheres, and then passaged them several times, then prepared a single cell suspension, and then transplanted into the hippocampus. Then 2 months after transplantation, we analyzed these animals for different behavior. Particularly, we examined depressive-like behavior using a forced swim test, and also we tested their ability for recognition memory using a test called novel object recognition test, and then spatial learning and memory using a standard water maze test. Now, properties of these cells in culture. So I mentioned earlier that these cells express a variety of neurotrophic factors. In this picture, you can see these cells express BDNF, GDNF, as well as VEGF here, and some of the cells also express FGF2. And if you maintain in culture, especially if you plate them on substrate-coated dishes, they differentiate into neurons, astrocytes, and oligodendrocytes, and you can see the 2J1 positive neurons here, and majority of them are also GABA positive. And if you wait longer, in long-term cultures, you see actually subtypes of interneurons expressing calcium-binding proteins such as parvalbumin, calretinin, calbindin. And then you also see some interneurons expressing somatostatin and neuropeptide Y. Here in this picture, you see a differentiation of these cells into astrocytes. Mature astrocytes are identified with the marker called S100 beta, and then oligodendrocytes are identified with O4 as well as RIP. It shows the percentage of different types of cells, and you can see there over 20% of cells differentiate into neurons here, and then most of them are GABAergic. Then over 60% of cells are GFAP positive. This is a mixed population of cells because GFAP is expressed in astrocytes as well as undifferentiated stem cells. So that's why we also quantified the percentage of S100 beta positive cells. About 50% of them are S100 beta positive, so they are actually mature astrocytes. Then oligodendrocytes expressing O4, about 15%, and RIP is the other marker of oligodendrocyte that also shows about 15 to 20%. So these are really neural stem cells because they can give rise to all three CNS phenotypes: neurons, astrocytes, and oligodendrocytes. Okay, now coming to the effects of these transplants on behavior. So the first test was a forced swim test. This is a test for depressive-like behavior. And this is a simple apparatus here. It's a large beaker filled with water. You just place the rat in that. They don't like to be in the water. They struggle a bit initially, but control rats swim continuously after that. Only when they get tired, they take little breaks. They exhibit a behavior called floating behavior. It's also called immobility. So that involves minimal movement of limbs. So the extent of immobility determines the extent of depressive-like behavior. So depressed rats, they give up swimming faster. They spend most of the time in floating. So we basically measure the amount of time spent in floating. You can see here, control rats, the blue bar here. So they spend less than 50 seconds in floating. And this test lasts for 10 minutes. So out of 600 seconds, only 50 seconds they spend in floating. Whereas the animals with hippocampal injury, the red bar there, so they spend more time in floating. So implying that they have increased depressive-like behavior. But with transplantation, it is normalized. So it's very close to control values here and much less than the time spent in floating by rats with injury alone. So it indicates that neural stem cell grafting intervention early after injury, it prevents depressive-like behavior in these rats. So then we did a novel object recognition test. This is another simple cognitive test. It involves two major phases here. In the first phase, the animals were allowed to explore two identical objects for about 5 minutes, and then they're taken back to their home cages, and 2 hours later, we'll place them in the same open field, but one of the objects is replaced with a new object. So it's a natural tendency of rats to spend more time with the novel object, and that involves the learning and memory process. So basically what we do is in the testing phase, we quantify the percentage of time spent with the novel object. So control rats, since they remember very well, they spend nearly 70% of their object exploration time with the novel objects, indicating that they have very good ability for object discrimination. Whereas the animals with injury, they spent less than 40% of their time with the novel object, clearly indicating that indicating their inability for object discrimination. But transplanted animals, after injury, they did much better, and their object exploration time was novel object exploration time was closer to control values. So it implies that neural stem cell grafting early after injury actually preserves recognition memory function. So next we performed water maze testing. It's a simple water maze test involving 7 sessions of learning over 7 days. The animals were, they were supposed to find the hidden platform. In each session we had 4 trials. And this shows the learning curves for the 3 groups of animals. The first one is for the control group. This is for the injury alone group. And this is the injured group with transplants. As you can see, the learning was not different between the 3 groups. So the partial hippocampal injury does not interfere with the water maze learning. But when we performed a memory test, so this is also called a probe test. This is typically done 24 hours after the last learning session. So what we do is we take out the platform, and then each animal is given a single trial of, it lasts for about 45 seconds. So animals, if they remember, they head straight to the quadrant of the pool where the platform was originally placed, and they start searching for the platform. So eventually they spend more time in the platform quadrant. So that's why we quantified the amount of time spent in platform quadrant as well as in the platform area. That area denotes the position of a platform as well as immediate surrounding region. As you can see here, control rats, they remember really well. They spent a greater amount of their probe test time in the platform quadrant as well as in platform area, whereas animals with injury, they spent a reduced amount of time in the platform quadrant as well as platform area. But transplanted animals did much better, and their values are closer to control values, implying that so they have a normal memory function. So again, so grafting early after injury prevented spatial memory dysfunction. So then we looked at the distribution of transplant-derived cells. I mentioned earlier that these transplanted cells are AP positive, alkaline phosphatase positive, but they were also labeled with BRDU because we added a small amount of BRDU to neural stem cell cultures. So every cell got labeled with BRDU too. So we quantified the numbers of BRDU positive cells in different regions of the hippocampus, and using neuralistic, we also mapped the location of these cells. And overall yield was equivalent to 116% of injected cells. So we got more cells than what we injected. This is partly because some cells actually divided after transplantation. And cells also migrated into different regions. The core is in the CA3 here. This is at higher magnification showing CA3, and then you can see in the dentate hilus, many cells are migrated. And in this picture, you can see a lot of cells have gone into the subgranular zone and granule cell layer of the hippocampus. So based on our quantification, about 20% of the cells, they migrate into the granule cell layer or the neurogenic niche, and remaining 80% of cells, they are distributed all over the hippocampus. Then we looked at the phenotype of transplant-derived cells using dual immunofluorescence. In this case, we used alkaline phosphatase and NEWN, and you can see a large number of NEWN positive neurons. This is in the core of the transplant, and a majority of those NEWN positive neurons are also positive for GABA, when we performed alkaline phosphatase and GABA immunofluorescence. And then we also looked for subclasses of GABAergic neurons, and many of them differentiated into subclasses expressing calcium-binding protein calbindin and neuropeptide Y, and as well as parvalbumin. And then we also looked for other cell types, glial cell types. In this case, we did S100 beta to demonstrate the presence of astrocytes among graft-derived cells. And then we used CNPAs for identifying oligodendrocytes, and then NG2 for identifying oligodendrocyte precursors. And here you see percentages of different types of cells. Close to 30% of them are neurons, and majority of them are GABA positive. And then we also found significant numbers of calbindin and PV. NPY neurons are occasionally seen, so that's why we didn't quantify that. And S100 beta, nearly 50% of them are astrocytes. And then we found about 15% of cells differentiating into NG2 oligodendrocyte precursors, and some into mature oligodendrocytes expressing CNPAs. Okay, then we looked at the expression of neurotrophic factors in transplant-derived cells. Particularly, we focused on factors that were expressed at the time of transplantation. So GDNF, you can see there, the transplant area shows excellent GDNF expression. One interesting thing is in the chronically epileptic hippocampus, there is significant downregulation of GDNF in host astrocytes. But in transplanted astrocytes, transplant-derived astrocytes, we see a lot of GDNF. So nearly 50% of transplant-derived cells express GDNF. So GDNF is an important protein because it also acts as an anticonvulsant factor. Then we found expression of BDNF, FGF2, as well as VEGF in different cell types. So close to 50% of transplant-derived cells expressed a variety of neurotrophic factors. Then we looked at the effect of transplantation on neurogenesis because of the linkage between neurogenesis and learning and memory as well as depression. So the first panel here shows control hippocampus, age-matched control hippocampus. You see nice-looking double carotene-positive neurons with the normal orientation of dendrites into the molecular layer. And this is the chronically injured hippocampus without transplant here. And you can see neurogenesis decreases there. And then this is the transplanted hippocampus. And when we counted double carotene-positive neurons, we found similar numbers of double carotene-positive neurons between control animals and transplanted animals, but significant decrease in the number of double carotene-positive neurons in injured animals that did not receive any treatment. Surprisingly, we also found some increase in neurogenesis in hippocampus contralateral to injury. We do not know why that happens, but we do see a significant increase in neurogenesis after transplantation into the injured hippocampus. So then we looked at abnormal neurogenesis. Steve has talked a lot about the basal dendrites and abnormal migration of newly born neurons into the dentate hilus. So we wanted to see whether early intervention with neural stem cell grafting, can we prevent the formation of basal dendrites from newly born neurons? And can we prevent the migration of newly born neurons into the dentate hilus? And the answer is yes. As you can see here, we quantified the percentage of double carotene-positive neurons in the dentate hilus expressing, this is the percentage of neurons in the dentate hilus. You can see that injured hippocampus, there's a significant increase there compared to control. And in transplanted, it comes down. It's not as close to control, but much less compared to the animals with injury alone. And then we also quantified the numbers of newly born neurons with basal dendrites. Again, with injury, it increases significantly. Then with transplantation, it's normalized. So clearly, the transplantation decreases the migration of newly born neurons into the dentate hilus and also decreases the formation of basal dendrites. And we wanted to know why this happens. So there is some connection between reelin and abnormal migration of newly born neurons, originally shown elegantly by Jack Parent. So reelin-positive neurons, you see a large number of reelin-positive neurons in the dentate hilus, and they are playing a major role in appropriate migration of newly born neurons into the granule cell layer. So when you lose a lot of reelin-positive neurons, such as after status epilepticus, then the guidance is not there. So instead of going into the granule cell layer, a lot of these newly born neurons, they take a detour and get into the dentate hilus. So in this case, we found decreased number of reelin-positive neurons with injury alone, but transplantation after injury rescued reelin-positive neurons, as you can see from the data here. So the part of the mechanism is that we are protecting reelin-positive neurons in the dentate hilus, and that reduces the abnormal neurogenesis. We also looked at the activity of stem cells. In this case, we did triple immunofluorescence for GFAP, Ka67, and S100 beta. But in this picture, you see only GFAP and Ka67. So the cells in the subgranular zone that are positive for GFAP but negative for S100 beta, they are the actual stem cells. So we quantified such cells with Ka67. We found that 15% of stem cells in the subgranular zone actually proliferate in the control hippocampus, but that goes down in the injured hippocampus to about 6%. But with early transplantation, we see about 12% of cells proliferating. So transplantation actually normalized the proliferation or proliferative activity of endogenous stem cells. So now major conclusions from this study is that subventricular zone neural stem cell grafting early after injury counteracts injury-induced mood and memory dysfunction. And preservation of function in grafted animals is associated with excellent yield and migration of graft-derived cells, differentiation into various subtypes of GABAergic interneurons, astrocytes and oligodendrocytes, and a significant number of graft-derived cells express neurotropic factors. They are very useful neurotropic factors, GDNF, BDNF, and FGF2, as well as VEGF. And we also found normalization of the extent as well as pattern of neurogenesis and protection of renin-positive interneurons in the dentate iris and increased neurogenesis in the hippocampus contralateral to injury. Okay, we did not study the effect of transplants on seizures in this model because it's a focal injury model. As I mentioned earlier, to see seizures in this model, we had to wait at least 4 months. But most of our studies were done before 3 months after injury. That's why we shifted to a different model and asked the same question: can neural stem cell grafting early after status epilepticus ease chronic epilepsy and mood and cognitive dysfunction? So we used a status epilepticus model. As you know, status epilepticus is a life-threatening emergency typified by prolonged seizure activity. The incidence is 60 cases per 100,000 population, and overall mortality is about 20%, and causes are many. It can result from stroke, trauma, infections, tumors, drug withdrawal, Alzheimer's disease, as well as ongoing epilepsy. And the available treatment is basically a bunch of anti-epileptic drugs. A lot of neurologists here, and they can tell more about that. The goal is to quickly terminate seizures. The consequences, anti-epileptic drugs, they stop seizures, but do not modify the detrimental effects of status epilepticus. The detrimental effects include development of chronic epilepsy and comorbidities such as depression, memory dysfunction, and then impairments in hippocampal neurogenesis. This is a cartoon. It shows some of the major events after the initial precipitating injuries such as status epilepticus. You see bilateral hippocampal neurodegeneration. So both sides you see loss of neurons, principal neurons as well as interneurons. Then you see abnormal neurogenesis, reductions in different subclasses of GABAergic interneurons, and abnormal synaptic reorganization such as mossy fiber sprouting. And all of these contribute to hyperexcitability, which eventually leads to chronic epilepsy typified by spontaneous recurrent seizures, depression, cognitive dysfunction. Okay, this is the animal model we use. This is the kainate model of temporal lobe epilepsy. So what we do here is animals receive graded intraperitoneal injections of canic acid, originally developed by Ed Dudek for Sprague Dawley rats, but we modified it to Fisher 344 rats. Since Fisher 344 are more susceptible to seizures, we had to reduce the dose a lot here. So we gave 3 milligram per kilogram body weight for 3 to 4 hours. So it's hourly injections, and you give injections until the animals develops status epilepticus. So once they develop status epilepticus, you see continuous stages 3 to 4 seizures, 3 to 5 seizures for over 3 hours. Then we give an injection of diazepam to terminate the behavioral seizures. Then a few months later, if you observe these rats through direct observation or video recording, you see spontaneous recurrent seizures. We quantify those recurrent seizures and then identify animals with chronic epilepsy. This shows some of the EEG activity during status epilepticus. There are different stages. You see initially wet dog shakes, and then after that you see only electrographic seizures for a while, and then you start to see behavioral component of seizures like stage 3 seizure with unilateral forelimb clonus, bilateral forelimb clonus, that's stage 4, and then stage 5, bilateral forelimb clonus with rearing and falling. Then this shows electrographic activity during continuous seizures. Cell death, I mean, many people have shown this. Status epilepticus kills neurons in the hippocampus in several layers. With fluorogate, you can see loss of neurons in dentate hilus, CA1, and this is in CA3, and also in extrahippocampal regions like piriform cortex and entorhinal cortex, you see cell death. And this is, upper panel is an example of milder injury at 48 hours after the induction of status, and the lower panel is an example of severe injury. Of course, you can also see dying neurons if you do silver staining. So in this case, we identified degenerating neurons through silver staining. And some of these cells die through apoptosis. If you do tunnel staining, you can identify that. But cell death is not just apoptosis after status. Some cells also die by necrosis. And if you wait 4 months, you see the hippocampus like this. The upper panel is control. The lower panels are the examples of animals that underwent status. You see significant loss of neurons in the dentate hilus, and then also CA1 cell layer partially gone. CA3 cell layer is partially gone in some areas. So the type of injury is quite variable, but most animals have loss of neurons in CA1 and CA3, as well as in dentate hilus. That's what, when we quantified new and positive neurons, we found decrease in dentate hilus, CA1, as well as in CA3, but no significant change in dentate granule cell layer. That's partly because there is increased neurogenesis that probably compensates for some of the loss that's occurring due to status epilepticus. So then it shows neurogenesis. I'm not going into details here because Steve has shown very extensively the early phase after status, how neurogenesis changes. It's control hippocampus here, and this is 12 days after status. You can see a large number of neurons migrating into the dentate hilus here, and some of them have very clear basal dendrites. And when you quantify, you see dramatic increase in neurogenesis and also increase in the number of new granule cells in the dentate hilus. But in the chronic phase, just as in the other focal injury model, we see dramatic decrease in neurogenesis. So this is control hippocampus, and this is the chronically epileptic hippocampus 5 months after status. And when you quantify double cortical neurons, we found significant decrease there at both anterior and posterior levels. And whatever residual neurogenesis you see, that is completely abnormal, because most of the newly born cells, they get into the hilus or they send basal dendrites into the dentate hilus. We also looked at loss of interneurons. In this case, we concentrated on neuropeptide Y because it's highly vulnerable to status epilepticus, particularly in the kainate model. And PV, as you can see here, in different regions of hippocampus, both of these populations decrease in number. So this shows the experimental design for transplantation study. On this side, you see the preparation of donor cells here. So we dissected subventricular zone tissue and then expanded them, neural stem cells as neurospheres, and then prepared a single cell suspension and transplanted. On this side, you see the preparation of rats. So we induced status epilepticus, and they had seizures or status epilepticus for 3 hours. Then we terminated seizures with diazepam. And 4 days after the induction of status, we classified them into 3 different groups. The one group received transplants, and in this case, we bilaterally transplanted neural stem cells. Each hippocampus received 4 transplants containing about 90,000 live cells. And the other group received sham grafting surgery. So instead of transplanting live cells, we transplanted dead neural stem cells. And the third group was maintained as epilepsy-only controls. First, we quantified the progression of seizures in these 3 groups. As you can see here, the red bars, they denote animals that did not receive any treatment after status. And you can see increased frequency of seizures between months 3 and 5 after status. And in animals receiving dead cell transplants also, we found similar scenario, increased frequency of seizures at 3 to 5 months post-status epilepticus. But transplanted animals showed very few seizures. So clearly, the transplantation suppressed the frequency of seizures. We also quantified the frequency of the most severe type of seizures. There's a stage 5 seizure, typified by bilateral forelimb clonus, rearing and falling. As you can see there, there's a significant decrease in the frequency of stage 5 seizures in animals receiving transplants. We also quantified duration of individual seizures, and that is also decreased, highly significant in the 5th month after status. And then this is the percentage of recorded time spent in seizure activity. And you can see that there's a dramatic decline in the amount of time spent in seizures by transplanted animals. So clearly, transplant had a positive effect in terms of suppressing seizures after status. Okay, then we did EEG recordings. In this case, we did 24/7 recordings for 96 hours. And in this also, we found a significant effect of transplants on seizures, a 59% reduction in the frequency of all seizures This includes behavioral seizures as well as electrographic seizures. And then 97% reduction in stage 5 seizures, and 45% reduction in the duration of individual seizures, and 72% reduction in the percentage of recorded time spent in seizure activity. This shows a few examples of seizures. Generally, seizures are more severe in animals not receiving any treatment, but animals receiving transplants, when they had seizures, seizures were a bit milder compared to the epilepsy alone rats. So transplantation not only decreased the frequency, but also decreased the intensity of seizures during the chronic phase. So then we did the similar behavioral test, which I showed earlier. It's a novel object recognition test, quantified their ability for object discrimination. And you can see here the control rats, they have very good ability for object discrimination because they spend 80% of their exploration time with the novel object. But epileptic animals, they lose that ability, so they have recognition memory dysfunction. But animals that receive transplants after status, they clearly show ability for object discrimination. So again, so early neural stem cell grafting intervention after status prevents recognition memory dysfunction. So we did the depression test, the forced sim test, and quantified the amount of immobility time. This is in the first 5 minutes of test, in the second 5 minutes, total duration 10 minutes. With all of these quantifications, we found a very positive effect of transplants in terms of preventing depressive-like behavior in epileptic animals. So we looked at histology then. So this is a study in progress. So I have some data on the transplant integration. So whatever I have, I'm going to show today. So we did BRD immunostaining to identify the transplant-derived cells, and we found that recovery was equivalent to 89% of injected cells, slightly less than the other model, but still 89% survival or recovery is excellent. And in this model also, we found that about 25% of transplant-derived cells, they migrate into the dentate granule cell layer or into the subgranular zone. And that they differentiate into new and positive neurons. And all of these neurons, they seem to differentiate into granule cells or glutamatergic neurons, because when we did GABA staining with BRD, we did not see any GABA-positive neurons in the dentate granule cell layer. But the remaining 75% of graft-derived cells, they migrate into other regions of the hippocampus. And out of that, 24% differentiate into neurons, and then the rest into glia. And among neurons in other areas, nearly 22% of graft-derived cells differentiate into GABAergic neurons. Then we also looked for astrocytes expressing S100 beta, and we found that 60% of graft-derived cells differentiate into S100 beta positive astrocytes. We also looked at the effect of early transplantation on the survival of host interneurons. In this case, neuropeptide Y positive interneurons, control hippocampus, and this shows chronically epileptic hippocampus. You can see massive decrease in the density of NPY positive interneurons in the hilus, but with early transplantation, we could rescue NPY positive neurons. And they also exhibit hypertrophy. So most likely, these neurons are working much more than in the normal hippocampus. So you can see the cell body size is much bigger here. So this is the quantified data, and you can see clear decrease in NPY positive interneurons without treatment, but with neural stem cell treatment, you see much rescue here. And similar story for parvalbumin positive neurons. Status epilepticus leads to significant loss of PV positive interneurons in the chronic phase. This was examined 9 months after status, but with early transplantation, so you can see the rescue of PV positive interneurons. Then we looked at neurogenesis, again, very similar to what we found in the focal injury model. So neurogenesis was maintained in transplanted animals, but greatly decreased in animals that did not receive any transplants after status epilepticus. And we also quantified the number of newly born cells that migrate into the dentate hilus. Instead of using double cotton, here we used PROX1. So double cotton shows only the status of neurogenesis because it's expressed for about 2 weeks. So whatever neurons you see with double cotton staining, they are the neurons that are generated 2 weeks prior to euthanasia. But if you do PROX1 staining, you can identify all granule cells that accumulate over a period of time. So that's why in this particular study, we quantified the total number of PROX1 positive cells in the dentate hilus. And you can see here in animals that did not receive any transplant, it's about 50,000 granule cells accumulate in the hilus. But with early transplantation, that is reduced to about 30%. So clearly, early transplantations prevented the migration of newly born neurons into the dentate hilus. And again, we wondered whether it is due to the rescue of reelin-positive neurons. We did immunostaining for reelin, and that happens to be the case here. You can see the significant decrease in epilepsy alone rats, but rescue of reelin-positive neurons in rats receiving neural stem cell transplants. We haven't quantified this one, but this is a qualitative picture my postdoc sent yesterday, so I thought I should show this. Yeah. And we also looked at mossy fiber sprouting because of two reasons. One is, when animals have a lot of seizures, you see, significant mossy fiber sprouting in the dentate supragranular layer. And the other reason is based on Steve's and Jack Parent's data, and most of this mossy fiber sprouting comes from neurons that migrate into the dentate hilus. So as you can see, at the control hippocampus, you don't see any dentate supragranular layer because there is no mossy fiber sprouting in that area. But in animals that underwent status epilepticus, you see a very thick band of mossy fiber sprouting there in the dentate supragranular layer. So this was identified with the ZNT3 immunofluorescence that identifies zinc-containing axons. In transplanted animals, we did not see much mossy fiber sprouting. So this goes very well with the suppression of abnormal neurogenesis as well as suppression of seizures. Only in some cases we found a very band in the only in the lower blade of the dentate gyrus. But in most animals, we didn't see any mossy fiber sprouting after transplantation. So the transplantation also prevents abnormal mossy fiber sprouting. Now, to conclude this study, so neural stem cell grafting early after an episode of status epilepticus diminishes the frequency as well as intensity of both behavioral and electrographic seizures in the chronic phase. It prevents status epilepticus-related recognition memory dysfunction, prevents depressive-like behavior, preserves subclasses of GABAergic interneurons expressing NPY and parvalbumin as well as reelin, preserves normal pattern of neurogenesis. And functional and neuroprotective effects of these grafts were associated with excellent survival and migration of graft-derived cells, differentiation of graft-derived cells into neurons and glia, and then 22% of graft-derived cells differentiate into GABA. So that leads to significant addition of new GABAergic interneurons into the hippocampal circuitry. Well, the funding is mainly from NIH and from the Department of Veterans Affairs, and these are the people who contributed to this work. Thank you for your attention.